It's my pleasure to speak to you today at this exciting time for AdAlta as they transition their first product, AD214, into the clinic. I'd like to describe to you our clinical strategy for AD214, a program that's been really strongly underpinned by our preclinical data for AD214 and also has been informed by key opinion leaders in the therapeutic area of interest, the key therapeutic indication that we're targeting at this point in time, as Mick had introduced you, was interstitial pulmonary fibrosis. We are planning on incorporating a radio-labelled PET tracer, as Mick described, which enables us to collect data via PET-CT imaging in our patient cohorts, which provides additional insights into the target engagement of AD214, the biodistribution and pharmacodynamics as well. So even though the target indication for AD214 is planned to be interstitial pulmonary fibrosis, recruitment into the first study is going to be open to a broader range of diseases, interstitial lung diseases. Now this broader group of disease, all have within their pathology chronic fibrosis of the lung. First patient results are expected early in 2021. So a little bit more information on the proposed two-part phase 1 design. As I said, we're kicking off the study with a Part A single-dose healthy volunteer study. We plan to recruit up to 40 subjects in 5-dose cohorts ranging from 1 to 20 mg per kg IV. I'll discuss those doses that we've chosen in the next slides. The study is a strong randomized 3-to-1 ratio receiving AD214 or placebo. This enables us to rapidly transition into Part B of our study. Part B of our study, which is also a single ascending dose study in ILD patients, can begin after the 5 mg per kg dose is cleared in our healthy volunteers study. We are starting at a much lower dose of 0.1. Again, to provide additional safety data, and I can discuss those doses later on, and planning to escalate to 20 mg per kg IV. And we'll be recruiting from 2 to 3 sites to enable us to recruit and finish the study within our timelines. The single dose study will then transition into a multiple dose ascending dose study in ILD patients also. Again, at 2 to 3 sites, we aim to recruit 12 to 24 subjects over 4 dose cohorts ranging from 1 to 20 mg per kg. This study, again, can begin after we clear the 5 mg per kg cohort in the single ascending dose. So as with any first-in-human study, our initial and our primary objectives are to investigate the safety and tolerability of AD214. We have additional secondary objectives to collect PK and PD data for AD214 in both a healthy volunteer cohort and also in our patient population of interest. Our multi-dose study will also enable to collect information on the immunogenicity of AD214. I think the value-add objectives, because we are planning on recruiting patients, and our exploratory objectives are to explore the effect of AD214 on respiratory function in these patients, and also the inclusion of the PET tracer, as we discussed before, enables us to look at the localization and distribution of AD214 in the lungs of these patients. The patients that we're recruiting, obviously they have to meet a long list of eligibility criteria which will determine whether or not they meet the requirements of being a patient population with interstitial lung disease. And as Kevin had mentioned, interstitial lung diseases are a heterogeneous group of disorders which all have within their core, and this is very important, a pathology of chronic fibrosis in the lung. And also this group of patients is also known to have high CXCR4 expression. Now, idiopathic pulmonary fibrosis makes up roughly 20% of that patient population. The study will be open both to IPF patients and also to the larger group of patients who have clinical and radiological features of ILD. Now, the sites that we've chosen for this study and the PIs that we are recruiting for this study, the principal investigators, have got a lot of experience, and in fact, the broadening of our target population actually came on their advice because they felt that it was a relevant target population and also enabled us to complete the study within the timelines that we've set ourselves. So a little bit more colour on the reason that we've chosen our doses of 0.1 to 20 for our patient cohort and 1 to 20 for our healthy volunteers. So you'll remember, as Mick described, that we had escalated in our non-human primate model to 100 mg per kg. And at that point in time, there was no overt safety signals, and the dose was determined to be tolerable. Human equivalent dose of that dose of 100 mg per kg is roughly 32.3 mg per kg. Our 20 mg per kg dose is our highest dose that we've chosen, provides an extra 30% safety margin on that potential maximum dose that we can choose. And additionally, the 20 mg per kg actually sits quite well within what we know and what we know from what is currently used for other therapeutic proteins on the market. The other important thing to understand is that we've also chosen our starting dose of 1 mg per kg in our healthy volunteer cohort, and that's according to regulatory guidelines that suggest that the starting dose in a healthy volunteer cohort should be approximately 10-fold less than the human equivalent dose. And in fact, we've gone greater than 10-fold less because we're starting 30-fold less. So that provides additional safety for our healthy volunteer cohort. We have, on the advice of our key opinion leaders, again, these are respiratory physicians, in fact, interstitial lung disease physicians. Additionally, we have an extra 10-fold safety margin in our patient cohorts where we're beginning dosing of our patient cohorts at 0.1 milligrams per kilo. I think importantly, what we have to understand that this range of 1 to 20 and 0.1 to 20 is also what we believe to be a potential therapeutic dose, again, guided by our preclinical data. So you remember Mick described our occupancy data in our receptor occupancy data in our non-human primate model, where it was demonstrated that the 30 mg per kg dose dosed weekly gave greater than 60% receptor occupancy for greater than 3 days. The human equivalent dose of 30 mg per kg in the non-human primate is 10 mg per kg. Therefore, we are well within that therapeutic range, potential therapeutic range. In addition, our mouse bleomycin model data showed us that 10 milligrams per kilo was effective to prevent or to treat fibrosis in this animal model of fibrosis, and the human equivalent dose of that is 1 milligram per kilo. So with respect to timelines, we have already completed the 8214 drug product manufacturing run, which we have, as Mick had described, already begun development of the PET tracer. And we've also engaged our key vendors to start and begin the process of submitting or preparing the paperwork that enables submission of our protocol, investigators brochure and all of the data collection requirements for our phase 1 study, part A of our phase 1 study, which we expect to be dosing in the middle of the year. We expect that the patient cohort population will begin mid-Q1 with data readouts of 2021, with first data readouts in Q2. And then that will transition, as we described previously, quite quickly into the proposed multiple escalating dose component of the patient study. Now you'll note we do have the PET tracer development, so at the moment we're planning on submitting the protocol with the option of including the PET tracer in, so there will potentially be a protocol amendment that will be required to input the data from the development of the PET tracer at that point. All going to plan, we anticipate that we could be ready for phase 2 studies in Q4 of 2022. And what would a phase 2 program look like, I should say, for interstitial pulmonary fibrosis? If we have a look at a snapshot of what other studies have been completed in this space, Galapagos, Fibrogen, and other companies have completed phase 2 programs, and some have actually progressed to phase 3 programs in this disease. And a phase 2 program in IPF, basically all of these studies have shown that you would need to recruit between 20 and 100 patients. They have follow-ups ranging from 3 to 12 months, depending on what they're looking at. They tend to be open label without standard of care comparisons, and we anticipate that a program of 20 to 100 patients would be estimated to cost between $5 and $20 million. For an indication that has the potential to attract both FDA fast track designation and orphan designation.